Sasha, how are you?
Hi. Hi, how are you?
Hi, Sasha.
Hi. Very nice to meet you.
Great.
Yeah,
nice
nice
to
to
meet
meet
you
you
as well.
too.
Yeah.
We're still waiting for Christophe and Daniel.
We are.
Daniel, yeah, I was just on the phone with him. He's joining. Yeah.
Yeah.
Ah, yeah. Okay. Well, we'll maybe just wait for a few minutes and. Sasha, are you working exclusively for TSN or?
Well, I am retired. I'm not supposed to be working. Yet, like,
okay.
I am working.
You still look so young. Are you retired?
Well, it's Zoom. So, yeah, and no, I'm actually like independent. I don't work for TrialSite. Daniel invited me to write. I've been doing volunteer work for over a year. My background is in pharmaceutical research and development. I ran some clinical research organizations and I sold them. That's how I retired.
Yeah.
And and then I noticed what's going on, and I just couldn't. I was just like, okay, I can't sit and be quiet, and so I started. I started doing analysis, mostly like the VAERS analysis by lot numbers. That's where I started, and then started writing and publishing videos. And I'm doing right now. I'm mostly reading Pfizer documents and showing regulatory fraud. How FDA designed that strategy for them, and it's total fraud.
It's all unbelievable. It's all unbelievable. Yeah. But So I'm of the opinion that as from now it will all go so fast that I'm really convinced of it. I'm really convinced we are very, very close to the turning point. Really,
turning point. You mean in what?
In the virus.
In virus, yes. I agree with that.
Well, it's difficult to agree with that. There is actually very few people who agree with me. And why is this? Because it is so insidious. It is so tricky. Because right now, in fact, the vaccinees are better and better protected thanks to this famous infection enhancing antibodies. These infection enhancing antibodies, they prevent, in fact, infection of lung cells. Hi, Daniel.
Hey,
how are you doing? Yeah. Well, Christophe will probably join us in a minute, but I was just, we just introduced ourselves, with Sasha. And I was just, she was saying that she was looking into all this data, also not only the VAERS, but also the Pfizer data, and to see, how FDA played the game and just, kind of, approved everything, just playing their game basically. And I was just saying that I'm of course aware of all this analysis and even legal procedures going on and people doing lots of analysis. But I was just saying that the virus will hit us within a few. I don't know. I cannot say a few days, a few weeks, but I'm pretty certain it will still be this summer, right? And I was also saying very few people are actually believing me because the signs are not that negative. Of course, we get a lot of people vaccinees right now in the hospital, etc., but they come in with COVID, with SARS-CoV-2. So it's not their disease is, not due to COVID. It's not, due to SARS-CoV-2. It is like, secondary complications or other viral bacterial infections or even cancers and all this kind of stuff. So with regard to, if you look at hospitalizations purely due to COVID-19, they are low. They are low due to COVID-19, right? And that is because the antibodies that the vaccinees are right now developing, these famous infection enhancing antibodies that make them basically more susceptible to disease, right? Is protecting their lungs, so to say. It's protecting the infection of the lung cells. So that means that you don't see a lot of severe disease that is due to SARS-CoV-2 in these people. And on the other hand, the unvaccinated, their innate immune system get better and better trained, better and better familiarized with the virus. So also there is not a lot of hospitalizations due to severe disease going on. So, people get the impression, and even officials, that, we get a lot of other diseases, and now also with the monkeypox and avian flu and other infectious diseases popping up. But the COVID-19 is pretty silent, right? And but of course, what I'm studying is how the virus is evolving, right? And how this whole system, the silencing of SARS-CoV-2 right now reflects a huge immune pressure that is put on the virus, and we know that in vitro, the virus has already become more virulent, right? I mean, BA4, BA5 are more virulent. But if you combine these viruses with infection enhancing antibodies, you will see that the virulence is mitigated, right? But if you test them not in the presence of these antibodies, you will see that the virulence is basically increasing. So the virus has already become more virulent. That's for a fact. It's just that the infection enhancing antibodies can still bind to this virus for now. And as long as they do that, they can mitigate the virulence at the level of the lung. They can basically inhibit a process that we call trans-infection, which is the infection virus that is in fact tethered to dendritic cells. So, and there the virus is in fact transmitted or transferred from the surface of the dendritic cell to the susceptible lung epithelial cell. That is the process of transinfection, and these antibodies are preventing or at least hampering that process. But that supposes that the virus can still, of course, bind to these antibodies, right? And that is exactly what the virus is right now doing, is moving, away from the susceptibility to be bound by the infection-enhancing antibodies. So when that happens, it's like, you go up a very steep hill, and then at the end of the hill is the abyss, right? So it's not like this is going to be very, very progressive, that over weeks and months, you're going to see more and more people, vaccinees going to the hospital because of COVID. It's going to be very, very huge, right? And very abrupt. And I'm always saying people should have known me for many years to realize that I'm not the kind of guy who, enjoys making statements like this, right? And I think that is one of the reasons why we would like to have this meeting today, because Clas, you and I, we have already been talking about this. Many people are now basically the problem of the vaccine is, just in very, very simple words, is the following: that because of these infection enhancing antibodies, the infectiousness of the virus increases tremendously. So, these vaccinees are sitting on these high titers because they're boosted all the time by the circulating Omicron, of course, right? They're sitting on high titers of these infection-enhancing antibodies. And when these antibodies bind to the virus, they dramatically increase the infectiousness of the virus. That is what makes the vaccinees much more susceptible to infection, to reinfection, in fact. But that means this enhanced infectiousness means that in fact the virus blows right through the innate immune system, right? Innate immune system is basically completely sidelined. And there are, of course, a number of vaccinees that have been vaccinated much later on in the process, when the mass vaccination started, right? And these people had the opportunity already to be exposed to the virus, some of them several times even before they got vaccinated. So they had an opportunity to train their innate immune system. And in those people, I think there is still a reasonable, a substantial likelihood that even despite the fact that they have been vaccinated and that they have infection enhancing antibodies, that to some extent, the innate immune system is not completely sidelined because the innate effector cells have been trained and can still capture to some extent the virus and will induce natural immunity. So if that is the case, then we would see, in contrast to the situation with the vaccinees, where the innate immune system is completely sidelined, we would see in those people who still have some functionality of their innate immune system, that they have to some extent natural immunity, which would mean that they develop antibodies, not just against spike as the vaccinees, the people who are thoroughly vaccinated, they just, develop antibodies against spike, but the others would develop antibodies in principle against several different immunogenic components of the virus. So the most prominent of those are, of course, spike, but also nucleocapsid, for as far as I know. So I think because the question of, I'm receiving all the time the question of people, Geert, I'm vaccinated, is my innate immune system, is my capacity to generate natural immunity completely messed up or not, right? There is that one. There is the other one where young mothers, pregnant women, basically are told, get yourself vaccinated, because if you're vaccinated, you will have high titers of antibody in the blood, and that will basically passively protect through maternal antibodies the baby. Because, of course, it's like, the antibodies are transferred, and the reasoning is, well, these antibodies are, of course, infection enhancing antibodies. They still protect against severe disease. So in that way, you can protect the baby also against severe disease. And I've been making a number of statements, a number of calls over the last weeks, etc., talking to parents, video messages all over the place, how this would impact the education of the innate immune system of the newborn. But see, if mothers, for example, young mothers would now have the opportunity to get tested, let's say at an early stage of pregnancy or even before, and if they would find out that they have antibodies against nucleocapsid, against spike, so against several components of the virus, they could conclude that they have established natural immunity. And if during pregnancy they get exposed to the virus, that natural immunity will just be boosted, and the mother as well as the baby will benefit from it, right? So I think that there is a number of things. The focus class must from now be on serology, right? On serology. And also, people, for example, what my fear is, very seriously, I think that what is going to happen when the virus is now going to overcome this very last hurdle, where it simply does no longer bind to the infection enhancing antibodies. What we are going to see then is that we will see, antibody dependent enhancement of disease, because the virus will remain highly infectious. And since there is no protection left, because these infection enhancing antibodies will no longer protect against virulence, the virus is going to blow not only just through the innate immune system, but also, through the adaptive immune system. And then it may become extremely, extremely difficult for doctors to still treat these patients, because it will be really an exacerbated form of disease, will go very, very fast. I think there at a very early stage, we could have a test where we can still detect whether these people have some antibodies, against spike and nucleocapsid, that they have some natural immunity left. I think that may be a very important prognostic sign, and even an indication as to whether or not it makes sense to treat these people. Because I'm not a doom preacher, not at all. I'm expecting that when this happens, we are going to lose thousands and thousands of people, right? So we will need to select whether it makes sense to treat people or not, Because if they have still some natural immunity left, and now we come to your point, Clas. I saw your email from last night, and you treat these people immediately. You treat them immediately, and you can stop the viral disease. Then you will promote this little bit of natural immunity to further expand and to further develop, right? And that may be life-saving. So I think there is at least, 3 indications for this type of serology. But very seriously, we are not talking, I've been in the business as well, of course. But here, the exceptional thing is we are not talking about something to be developed and to be on the market in like a year from now. I think it's super, super urgent. We may even, with all our best efforts and everything, we may even come too late. But I think it's going to be a very, very important essay. And, my education is veterinary medicine. We do this all the time. We make tests, we do serology where we can discriminate between, for example, animals that have been naturally infected versus animals that have been vaccinated, etc. Because, we are always, with livestock, we are not thinking about the individual animal. We are thinking about the herd, the population, for economic reasons, of course. And, sometimes a whole population of animals is simply eradicated, right? I mean, this culling, we call this culling, right? When they test positive, for example, right? For economical reasons, you cannot do this, for example, of course, in human medicine. But I'm just saying the serology is now becoming so important to verify whether people still have some natural immunity left. Because remember, some of these people are vaccinated like 2 or 3 times. But very often, I'm coming from the vaccine field, there is no vaccine take. The vaccine take means that, these are non-responders. Some people are non-responders. They got vaccinated 2 or 3 times. They barely develop any antibodies because for some reason there has been a poor vaccine take. These people may panic, may think, they got vaccinated, they got boosted, and they are just going to fall victim of this very dramatic mutation. And it may not be the case. And one could easily test this, right? I think unless my reasoning is completely naive, and I'm not saying it's all very, very straightforward, but the point would be really to discriminate between the people who have no natural immunity left and those who have still natural immunity capacity left, because then we can build on this capacity. We can still try to expand this. Of course, some of these people will still get severe disease, maybe, but still treatable. Whereas I think that those who are really having no natural immunity left, the innate immunity, completely sidelined.
So Geert, if I could just chime in for a second, what's interesting is we just published a study yesterday. It hasn't been peer reviewed. It's a group of physicians in the Central Valley of California. Practitioner.
Oh yeah, I'm back. Okay.
And what they found, they studied the UK data recently with Omicron, and what they found are people are dying at an advancing rate and they're fully vaccinated, and their recommendation is, and I'm going to quote, that because of what's going on here, that we're going to have to take uniform screening protocols and protective measures irregardless of if one's vaccinated or not. So the point is that, and it's especially the elderly, are highly at risk right now. And so you have to be able, and so anyway, I just put that in the context that this is sort of validating in some way what you're talking about, because this is very recent and it's already been rejected by one journal, big surprise. But anyhow, sorry.
Yeah, all this stuff about the media and what people are saying and what public health authorities have in mind, and not forget about this. Really, it will go so fast. Forget about this monkeypox, forget about the avian flu. These are pandemics that are developing, of course, but the COVID-19 will, in highly vaccinated countries, the further evolution of SARS-CoV-2 will be so fast, And it will be so dramatic that, the other pandemics that are now developing, and by the way, only developing rather mild symptoms, right? That's going to be peanuts, right? That's going to be peanuts.
So on that note, Geert, and Clas, I wanted to reach out to Sasha because we've been collaborating on some other stuff, and Sasha has a significant background in building businesses around diagnostic technologies. Okay, and so I put that out there. Sasha, thanks for joining. Very grateful for your time. And so with that in mind, Geert and Clas, I mean, you have this test, and Sasha, maybe if you could talk a little bit about high-level best practices in business and diagnostics. I mean, there's broad, you can do everything, or you want to be focused with certain business use cases that are sustainable.
Yeah. I'd want to ask more questions, understand your test a little bit better. But in general, yes, so my background is I built 3 companies. We sold them. Well, we're still currently, I'm on the board of a medical device company, but it's in ophthalmology. So it's a different area, and it's not diagnostic, it's medical device. But previously we were working in various diagnostics, not blood related. So it was all digital data such as imaging, MRI, CT, X-ray, even photography, and then later electrophysiology, so electrocardiograms, but they're also digital and it's continuous, and 12-lead electrocardiograms, and then Holters and the Zio patches and all kinds of things. And so the labs I built, and we went in the central lab direction. We tried to do diagnostic applications for healthcare practice, but we quickly realized that it's a very uphill battle if you want to get reimbursed by the insurance, because the requirements, and they became even worse. So the requirements to run prospective outcome trials, and I mean, just even get, there are various pathways you can get. It's first like experimental technology codes, and they seem to be okay, but that still takes a lot of time and money. But then after they give you those, they require you to run prospective outcomes trials. And then what happens, it happened to a lot of our competitors. They initially came out and they had really generous reimbursement codes, and the companies went through the roof. And then the Medicare, Medicaid, they see that, and physicians start using. So when nobody's using them, they give you generous code. When physicians start using it and it creates uptake, they quickly, the bean counters realize, oh my God, we're getting charged a lot, and they cut it extremely substantially. And then, so you spend all this money and all this investment doing it. And so that's why a lot of investors exited that, because the financial people realized quickly what's going on. So if you have something that nobody's using, you're going to get good reimbursement code. If you have something that a lot of people are using, the Medicare, Medicaid is going to come in and cut it.
Yeah.
So since then, the preference in the financial space, like investors, for out-of-pocket pay models, subscription models, or some other mechanisms, and they're not very happy about if the company says, and then the insurance will reimburse it, unless it's something that's already being used, and you can apply some sort of already existing code, and but that's another conversation. But so we decided early on, we tried to do sort of screening, especially for electrocardiograms, which is now kind of coming into play because of all this SADs, and they're saying, we should do the ECG screening and see if you have prolonged QT. Maybe you have predisposition to the sudden cardiac death. Anyway, that's what we were trying to do a long time ago. And we decided to go into the pharmaceutical research and development because they needed the same tests for clinical trials. But they have different requirements. It's not like for practice of medicine, it has completely different precision, reproducibility, and false positives, false negatives requirements, which we have created, and that required the central overread of the data. So we did that, and that would be sort of the central lab digital model where we would standardize equipment in the field, like at the clinical sites, and then they collect the data and send the data to us, and in a standardized protocol, and then we would collect the data set and then do the processing and analytics on it. So that was my experience. But I would like to understand more about the tests, how you planning to implement them, what are they saying, and also, what I've seen, you can say, well, it's applicable to everybody, but maybe it's worthwhile thinking about, what's the first group that you will go after, what's the target, and those should be the ones that have the highest need, and not only the highest need, but also that you can do something about it. So once you have the test result, then so what? What decision can be made, or intervention, or what the doctor can do, or and the third thing is thinking about the payment structure. Unfortunately, a lot in medicine is driven by making money. Actually, almost all of it. And that I'm talking about the doctors. They want to think of themselves as life-saving individuals, but in reality, they're business people. And working for a large corporation. So that's why you kind of have to think about cash flows, who gets what. Your technology is not going to be taken up by anybody unless they're making money. So that's the third thing you have to think through. Who makes money in what way, and who doesn't make money if your technology is on the market, because they will be preventing you from getting there.
So, Sasha, before we go to Clas, because we of course need to give Clas the opportunity to react, what I would like to emphasize is that, I'm absolutely convinced that we will need to think out of the box, because, not be able to rely on established structures, the medical community or insurances or whatever. I'm convinced that we will be experiencing, the most unique, but of course in a negative way, the most unique moment that has ever been reported since the existence of man. I'm very serious about it.
Yeah.
So forget about using established structures, etc. I think there is only one thing that is the demand, will come from the people. We will need to raise their awareness that there is a chance for them to get tested. And depending, of course, on what this test will cost and how user-friendly it is, I think we absolutely need to avoid that we engage in a kind of well-established structure where we need to go from A to B, from B to C, etc., before something gets approved, before it gets implemented, etc. There's no time for this. And I'm telling you, what will happen, the first thing that will happen is that the hospitals will crash. When that happens,
they're already crashing.
No, but this is still, I'm saying this is still, a little bit of a prelude to the whole thing. It has not started for me. Even what you're alluding to, Daniel, these are very often senior people, that where you were saying that get hospitalized and that vaccinees. Yes. Very often senior people that have now come to the clinic, and because they are, as vaccinee, they are repeatedly reinfected with the virus. Their CD8 T cells get depleted, right? Get exhausted, the immune system get exhausted. And because of that, they start, having, secondary infections. Of course, the virus, if the virus is around, for example, and sucks the CD8 T cells, you can have bacterial pneumonia, for example, right? So it's bacterial pneumonia. It's no longer viral pneumonia like with Delta, etc., that these people are diagnosed with. None of this stuff is what I'm talking about. I'm really talking about a huge wave, let's be very clear, a huge wave of severe disease hospitalizations and deaths, right? And I mean, believe me, you don't have to believe me, but I mean, that is the scenario that I want to, put forward, and to see whether within a context like this, we can still do something with, the serology and the kind of technology that Clas has been developing. So over to you, Clas. Sorry,
Yeah, let's see what the technology is, and then I can come back to that. Yeah.
Yeah, so thank you, Geert. I was hoping today's talk was actually going to be about, the Save Your Children talk. Maybe we can go back to that and do a little bit of a deep dive into the immunology behind it. But if we can kind of do like a business school 101 situation, problem, solution type, approach, is that a good friend of mine always said, seek first to understand.
Mm-hmm.
So no matter what's going on in the ER or in the clinic, it always starts with proper diagnostics.
Mm-hmm.
We need to know what's going on the ground, what's actually happening in the patient, because there's a lot of similar symptoms, across these. And there's also co-infections happening around the world. So we need to really understand what's going on with the patient, first of all. Second of all, we need to collect that data and we need to present that data. So we're actually developing, or we've been developing what we call the open bio. So it's basically a database and apps. It's kind of like the Google App Store for biology in a sense. So what we have is an instrument. My background is PCR development in a vaccine company. So I used to work in veterinary medicine, just like Geert. We're developing tests for salmon and various fish and poultry and things like that to look for disease using PCR. PCR is not suitable for serology testing, and it also has the problem that you cannot do it in the field. And we failed trying to do PCR in the field because every test is positive at a farm, basically. So what's important is so we came up with, the solution is to use lateral flow assays. Lateral flow assays can be used for both serology testing, so antibody testing, and antigen testing, both. Plus, you can use chemical and biological markers like water quality and food quality, things like that. So the technology is available from many manufacturers around the world, so it's scalable, right? So every country has a set of manufacturers that make their own lateral flow assays. India does, China does, the US does. So there's lateral flow assays everywhere. So what we're trying to do is standardize those tests and be able to read them with our machine. We've developed a universal lateral flow assay reader. The advantage of doing that is that you can get better sensitivity and you can get the data into a system very quickly. We also eliminate the human error by actually having to mechanically read the test with glasses and face shields and PPE and actually get that data right into the database. And you can get metadata from the patient, such as vaccination status and disease history and symptoms. So our technology is basically a lateral flow assay reader and the infrastructure to collect and distribute and analyze that data. There is lateral flow assay vendors around the world, in Taiwan, India, Brazil, and the US to develop tests for particular diseases. We've worked on malaria, dengue, chikungunya, hepatitis, HIV, both antigen and antibody testing. So we're bringing every day, we're just bringing more and more tests into the ecosystem, basically. Yeah.
So the lateral flow assay reader machine. So this is you running it as a central lab, or is it going to be somehow distributed?
Yeah. So through distributors, we basically can use this at so in Southeast Asia and South America, would be like the pharmacy setting, or at a doctor's offices could be getting the results right away instead of having to send samples into a central lab. Yeah, so basically the future office of the future doctor's office is where you come in and do a series of tests, either from blood, saliva, gum scraping, a nasal swab, nasopharyngeal swab, whatever it takes, whatever, it depends on the assay to do the collection. And you put it onto the assay, and then you let the machine read the assay within 15 minutes, and the patient will then get the data on the app immediately. Yeah, so it's a way to do analytics very quickly. Now, just skip on the just back to the technology real quick. So I used to design PCR machines, and at the core of a PCR machine is actually a camera and a light source. So it's the opacity that changes of each sample well within the PCR matrix with all these little sample wells. So that reaction actually causes a change in opacity inside each sample well. So you detect it with a camera and a light source, or a light source and a camera. In ELISA, which is used for serology testing, it's the same thing. You have sample wells where there's an optical change to see if there is a reaction taking place. So, but then lateral flow assays are traditionally just read by eye, the typical pregnancy tests, right? The paper strips. So if we can just read those same reactions with a microscopic camera, with a good light source that have consistent, we're actually been able to do quantitative lateral flow assays.
Yeah, we might be able to help. I'm working with the optics, the main optics people in the world, so in my business. So we certainly jig something up.
Yeah. So that's as end result of that is that we can bring any kind of lateral flow assay from any vendor into the system very, very quickly, and we can do it remotely. So our machines are designed to be sent out. We have machines in Brazil and India and Singapore and Australia and everywhere. So we can just basically put those in the lab. So what we discovered too is if you try to get test kits shipped all over the world, they get stuck in customs, they get stuck at the FDA, they get stuck at So it's basically you develop the test, you send the machines out, you develop the test at the laboratory of the vendors in that country. And that's more scalable, right? So you can just quickly spin up lateral flow assays for any kind of disease that comes up. And so that's where we're at now. We're just basically bringing more and more tests into the system. Hepatitis, of course, HIV, monkeypox is coming, and various serology tests for even pregnancy, beta hemoglobin for to look at diabetes, early diabetes, onset of diabetes. So there's it's basically a universal diagnostic instrument where you can bring the assays into the system very quickly.
So, and how big is the machine, and how much does it? So you're gonna sell the machine to the doctor's office, for example?
So it's exactly. It's through a distributor. So it's the size of it's just very small. It's just
okay, that's good.
Yeah, very small. So it's just designed to make a reader. And it has a attachment for a monitor, so you can get a nice big view, like a microscopic view of the assay as it's being tested. And we also developed equivalence values. So it's basically, we use either PCR or ELISA to calibrate the lateral flow assays against either positive controls or actual samples, so that we can get equivalent cycle thresholds or ECT values for PCR. That means that lateral flow assays now can be read quantitatively. So you can actually track viral throughout an infection by using multiple tests throughout the histopathology of the disease. Same thing with antibodies, right? So it will be relatively useless for antibody testing to be non-quantitative, right? So you need to see the value and how they go over time. So that's what.
So, Clas, where do we? Where do we stand with regard to? Well, first of all, I would have a question with regard to. How many samples can we test? What's the time? How long does it take to test a series of samples? How many samples can you test, for example, per day? And then very concretely, do we have any of this development going on for SARS-CoV-2 with regard to serology, looking at spike and nucleocapsid?
Yeah, so that's a great question. So the reading of each assay takes around 2 seconds, 1 to 2 seconds, and the machine can process 500 samples an hour. It's typical for hospitals in Taiwan to have 2 machines side by side, so you can feed, you can kind of feed them back to back. So you can easily do 1,000 samples per hour, 2 machines. Then there's some barcoding system that you can link it to specimen IDs, particularly for hospitals. Or to the app. We've done Spike IG in the beginning. There's quite a few companies who have already built lateral flow assays for serology, so we've been working with them. SARS SC2 has been a very popular test around the world, right? What we do see is that governments are recommending not to test for serology.
Well, of course,
which is absolutely insane.
Why are they What's their strategy? Why are they recommending against serology?
They just simply don't recommend it. I mean, even like particularly in Taiwan, we have to work with private clinics because the official clinic by the NIH system, they just don't. The doctors are afraid to order those tests. They just can't.
But why?
Yeah, but also one of the other reasons is that it makes people aware that they are no longer capable of developing antibodies against anything else than spike, right? And that is also, a type of message that they don't want to spread, right? That it's because everybody is talking about increasing resistance, of spike, in fact, to the neutralizing antibodies. And then people realize, my gosh, I've been infected. I got, I got a disease, and I'm just developing antibodies against spike. Huge, and suppressing all other antibodies. So therefore, it's so discriminative. I mean, we never have this situation with vaccines that you have this kind of suppression, like of the innate immunity, and that on the other hand, the antibodies that you don't want to have are at extreme height because they're boosted all the time by the circulating Omicron. So here, for me, if it would be semi-quantitative, it would be, more than enough already. But Clas, with regard to nucleocapsid, is there any serology you're aware of that? I know that some people have been developing some of these user-friendly assays, where you could even do with finger prick, etc., test a droplet of blood, etc. But none of this is really reliable, sensitive enough, and plenty of false positive and false negatives. But if in this system, in the lateral flow assay, you would have. You could test both. Probably nucleocapsid is the best one. The second one to test, spike and nucleocapsid. And how long would it take to develop that?
Yeah, what we see, what the data suggests now is that we have test kits from multiple manufacturers that started developing test kits quite early, and those test kits are very targeted for spike antibodies, right? They call spike IG antibodies. We see that the data in those vaccinated, they test positive for spike IG post-infection. And that's for an epitope that is really no longer in circulation. So, like you said earlier, Geert, is that we really need to have multiple epitope protections from natural infection rather than artificial infection.
Yeah.
And,
but two proteins would be sufficient, right? I mean, really, the nucleocapsid is completely discriminative. So
exactly.
So we don't need to have, we don't need to test all the other proteins, which are very weakly immunogenic. But I would need to take a deeper dive. I've done this in the past. I don't recall this from the top of my head, but I think nucleocapsid is really the right candidate as a second one, Right.
And particularly because that domain of the nucleocapsid is less, it doesn't mutate like spike does, right? It's more stable.
No, it's not put under pressure. It's not put under pressure.
There's no pressure on it. Yeah, exactly. And also is that the antigen tests are typically done from nucleocapsid protein and not from spike. We did try to use some spike test kits early on, but those are hard to. They don't last very long. They denature quite quickly.
Yeah. So Clas, the other very interesting thing. The more I think about it, the more I think we should develop this because the great thing. Well, it's sad to say, but the vaccinees are reinfected all the time, right?
Yes.
Because they're very susceptible. So you don't have to wait till they get the next infection to verify whether they are developing antibodies just against spike or also against other components of the virus, because they are continuously, because of their high susceptibility. So whatever time point would do, right, to take a blood sample.
Yeah. So what we see is that when you do the traditional finger prick, it's not really sensitive.
Yeah, I know.
Detect low levels of antibodies. So what we really need to do is to do venous blood or proper blood draw and cells. You don't need to use ELISA, so it doesn't require it to be sent into a central lab. So I think the ultimate combination to get a good sensitivity is to do venous blood draw in a pharmacy setting or the doctor setting, and then do lateral flow assay so you can get the results right away, or within minutes of testing.
Yeah.
And then read it when because a lot of these test kits, they don't last very long, so you have to read them within 15 to 20 minutes. Otherwise, they start to bleed. You have color.
So, could frozen blood samples be used as well, or does it need to be fresh blood? Because so many vaccinees have been, people have blood samples around all the time, and
If you use blood that contains red blood cells, you need to use an anticoagulant, because otherwise the color interferes with the nitrocellulose paper. Yeah, and that reduces sensitivity. So the best result is to do it in from serum and just use a reagent.
But if you use sera, it could be frozen sera, right?
Sure.
Of course. Yeah. Okay.
Yeah, but the reaction needs to happen at room temperature.
Yeah. Well, Daniel, you had a question.
Yeah,
Well,
Daniel had a
and this maybe is a naive question, but I was just looking at this, and it's interesting that there's statements online that the serum antibody test cannot be used to diagnose current COVID-19 because an antibody test does not detect SARS-CoV-2. It's like a circular. It's just weird.
Circular argument.
And I'm, but I mean, if we look at high risk, let's say children, we talk about kids, okay, I want to go out, I'm a parent, I want to go get my child, let's say my child got vaccinated, and what Geert's talking about is happening in whatever form, so I go get this test, okay, then what? Okay, then, I mean, let's just talk about this a little bit. Let's say my child, the innate immunity is going down and I'm concerned, okay? What am I going to do about it as a parent?
Yeah, well, I mean, at first, what I think is going to be very important is, first of all, I mean, I'm afraid to say that it will come down to this kind of conclusion that, first of all, this test will be used to discriminate which people can you still treat and, which people basically have no natural immunity left. And so that means by the time the virus becomes resistant, even to these infection enhancing antibodies, they have no immunity left, right? So now on an individual basis, you could verify whether your child or yourself or a pregnant mother is part of this, part of the population that has no natural immunity left, right? And I think that is very, very important to know, because if you know that you're still eligible for early treatment, then I mean, there is the perspective is pretty different. If you don't have any natural immunity left, nothing prevents you from doing a second assay, for example, a few weeks later. But if that is negative as well, being vaccinated and being exposed all the time, so to say, well, I think these people almost, when the wave starts, I think these people have only one chance, and which is almost prophylactically to take antivirals, right? Whereas this is important. Whereas if you do have some natural immunity left, you should not take prophylactically the antivirals, because you still have some natural immunity left. So you have to take that opportunity to train that again, to make that stronger. And there the treatment was what Clas was doing. And we know this very well from the veterinary field, because we would even on purpose infect animals, for example. If we don't have a vaccine, a commercial vaccine, we would infect animals. And by the time they start to show symptoms, we would treat them with antivirals. At that moment, they develop immunity, also, natural immunity, without being threatened by systemic or by severe disease, right? So that means these people who still have these natural immunity, if you know they have them and they develop the disease, you know that it will most likely not be really so exacerbated as somebody who has no immunity left. And then you can treat them. This will strengthen their natural immunity. So in one case, you have to take them prophylactically because otherwise, there is no defense left. In the other case, where there is still this natural immune defense left, you have, of course, to closely monitor them. When they start to develop symptoms, you treat them immediately. And they can strengthen their natural immunity. And that will need to be built up because they have a huge backlog. As unvaccinated, we have trained all the time. We have updated our innate immune system. They didn't have that chance because I was saying, due to the infection enhancing antibody, the virus is now blowing right through the innate immune system, right?
So basically,
is that answer your question?
I was going to say that, so especially, Geert, if there's an emergency, the countermeasure, I mean, an emergency of the magnitude that you're talking about, you better believe it, that creates the conditions for certain types of commerce. I mean, you still have to go through, the mechanisms out there, but the mechanisms change. But I think a key here is to have, evidence-based pathways that are developed, for these scenarios. That makes sense to me. Like, for example, if, your innate immunity is severely eroded, I mean, you want to know about this, and you want to be able to take measures to bolster grow that. On the other hand, to your point, if you have some left, then so this becomes a way to cope with this transition. Now, the current powers of medicine and regulatory are losing credibility. I mean, this is what's happening. So from a market business perspective, if you have the right people behind something like this, and they're vouching for it, and there's, the right regulatory, boxes ticked, I think there could be something here. I don't know, Sasha, what do you think?
So that was my next. It would be worthwhile because, I believe what you say, Geert, but we need evidence, right? So it would be worthwhile figuring out who are your key opinion leaders, doctors who you can give this technology to and try to have them stratify their patients somehow, like basically on running a small clinical trial this way, field clinical trial, right? So that would be first to collect some data so then you can publish. Now, what you're going to have, because you already told me, the powers that be are going to be resistant. So they're going to try to shut you down based on regulatory pathways. And so you need to figure out a way to not make those claims. So what you're selling a piece of equipment, and then the doctor does whatever he wants to do with that piece of equipment. So that's basically the regulatory pathway for you to avoid. So then, as long as you're compliant with the technical aspects of, making the machine and processing the test and all that, if you want to avoid making diagnostic claims, because that is going to box you into running a whole bunch of clinical trials for years. Okay, so as long as you don't make any diagnostic claims, you're just saying, I'm providing a tool to physicians. Physicians are allowed to make diagnostic claims themselves. And then, so through that mechanism, so you need to figure out that regulatory pathway to avoid those minefields, because and I know they want to prevent people from doing this, because eliminates this whole vaccine narrative. Remember, they're all obsessed about let's inject everybody, vaccine hesitancy, we have to overcome at all costs, even by killing children and pregnant women. We just have to overcome this vaccine hesitancy. And that is going to tell people, hey, you're vaccinated, you have no hope really. And these people with natural immunity do have hope. Right, so that's an awful message for everybody who's pushing the vaccine, because they're saying, hey, the vaccine is actually making things worse. So that's why they're going to come down real hard and going to say, and they're going to try to prevent you from doing it. And in the US, they have really very well developed mechanisms. FDA can come down really hard on making any marketing claims that haven't been approved with them through a prospective clinical trial. and then they can just ignore it in the case of vaccine. So, so that's why I'm saying, like, so you need to figure out some kind of a strategy where you make the physician the the diagnostic person. They make the diagnosis, you just give them tools. And the as much as possible off the shelf, approved already, things of that nature.
Yeah, but to your point, Sasha, in fact, here we are not really talking about a diagnostic tool, right? You could easily say you're just kind of like assessing antibodies, right? You're not saying, you're not saying you because the diagnosis is made, you are vaccinated, you're not vaccinated. You could test everyone, so you don't discriminate between people based on vaccine status. You don't need to do that. You don't say, you have this kind of disease based on your antibody test, etc. You're just making an assessment. You're just measuring, basically. Yeah.
Yeah, you're measuring, exactly. So what you do is provide a measurement tool to a physician who then makes his own decisions based on this. Now, I mean, of course, you'll have to have discussions with your KOLs, and these should be, sort of seminars, meetings, offline, where you develop, where you want them to tell you how they would use it. They want how they would use it, what decisions they would make, have them try it, and then give you feedback on, oh yeah, I've tried it with this kind of patient and this kind of, and I got these results, and then I did this, and that's extremely valuable. But that should be kind of a discussion between you and your KOLs. Now,
so far, I've been discussing this through email. We have not gone in detail, but they are very, very clearly aware. I've been discussing this with Peter McCullough and Pierre Corrie and also Paul Marek. Right. Because I'm telling them, guys, you have to be prepared to cases of really exacerbated disease that, even with your early treatment, you're not going to be able, and it would be, I mean, this is sad to say. For me, it's at this level, when it comes to the population, of course, with my veterinary background, I'm just thinking about populations very often. Well, it's not true. I care about the individual, of course, but I think we will come to a point where we will need to decide whom to treat and whom not to treat, right? And if there is no criteria for that, not a reasonable, medical rationale for doing that, then it becomes a complete mess, because you will invest many, many resources, time and energy and people in things that upfront don't make sense, right? And that will come at the cost of other people that you could still treat, and that will come at the cost of a huge burden, of course, on hospitals, on medical facilities, etc., etc. So
it's a delicate situation. So I mean, for the diagnostic test, you still want to have whatever your outcome, they still should have some sort of action that they can do. Otherwise, people don't want to take tests. If the answer is like, well, you can do it, that.
No, but I was saying those who have no immunity left, they need to take antivirals prophylactically.
That's a good message.
Look, this context. Unless we talk about really the individual, which would be, for example, the pregnant mother, right? She wants to know, and before she starts pregnancy or at the early stage of pregnancy, do I have natural immunity? But otherwise, when we talk at the population level, this only makes sense if the antivirals can come into the play. First of all, for people who have no immunity left, they need to take them prophylactically. And as I was saying, those who have immunity left, natural immunity left, in order to be able to train this, it's the infection and treat protocol. But they get the symptoms and then you treat them. But it's also with antivirals, of course, right?
Can I ask you about the antivirals?
The antivirals.
I have to. Sorry to interrupt. I have to run, but we'll keep talking. Klaus, great work you're working on. I mean, it's a very important area, and I think we're trying to, through Geert, bring some brain power to help you out. So we'll be in touch. Okay.
Thank you so much.
Thank you, guys.
Thank you, Daniel. Good to meet you.
Okay.
Yeah. So I had a question about antivirals, Geert. So are these existing approved antivirals, or are you looking at developing new, or perhaps a combination?
No, I mean the only antiviral. Look, Clas, we are talking again about a huge population here.
Yeah.
So you need to have something which is readily available. can be supplied in huge quantities at affordable cost and which is safe.
Yeah.
Now, how many antivirals do you know that respond to these criteria? I only know
Maybe 5.
1 or 2.
Yeah.
Eh?
Yeah.
No,
situation. Those are the same like AIDS cocktails, you know.
No, it's ivermectin and hydroxychloroquine.
Yeah, well.
But if you talk about large supply, affordable cost, readily available
exactly.
everywhere, you know, and safe, tested in millions of people.
Yes. Well, yeah, and that's where the uphill battle is going to be, because you're going to get the same resistance because of this. Because first of all, you're pointing out that natural immunity is better, and second of all, so that would prevent, let's say, a pregnant mother from getting vaccinated, which is what the government wants, the opposite. And then you have to think about the strategy of hydroxychloroquine, ivermectin, because you've seen how suppressed they are. Although in the U.S. now it's sort of okay, you can get them, you can get them prescribed most places, but other countries may be, and they may come back again and say, oh no, you're again promoting the horse-based and all that.
Sasha, I fully understand your point, but you see where I'm going. I am speculating. Well, it's not speculation. I'm assuming, that the attitude, when this happens, that the attitude towards all these things will dramatically change. And for example, it has already changed a little bit without these catastrophes happening, because right now it's still like very reasonable in the hospitals, etc. For example, towards natural immunity has been already a fight, going on everywhere, but it's no longer that strongly rejected and declined as it used to be at the beginning. And same for some of these antivirals, as you were just saying. I'm not saying they are becoming more and more flexible, but there is some, So when this happens and people realize that this is really the only way to save people's lives, I think, and if you have to wait for that moment till this resistance fully abates, and then develop the assays, I mean, it's going to be too late.
No, I wouldn't wait. I'm just saying, figure out a strategy to fly under the radar as far as possible before they realize what you're doing and start coming down hard.
Yeah, that's interesting.
I say, no, don't wait. Do it now. But figure out sort of.
Yeah, you would agree that we need to be ready, right?
Yeah, of course. Yeah, so the strategy. People, so I would really start. I don't know what the state of your technology, how ready it is to go. Let's say like tomorrow, I don't know, Pierre Corey wants to use it on his patients. Right. Are you able to send him something? Are you able to enable him to do it?
Yeah, so you come from a regulatory background, right? So regulatory-wise, we've passed the Taiwan FDA, which has a deal with the US FDA. So it's actually a class 1 device.
Oh, great.
And the test kits are class 2 device because they make a decision. So working with the test kits in type of very similar to BD Veritor, where they have a class 1, class 2 type device. We've also have approval from DCGI in India, and ANVISA in Brazil, and IVDR, IVDD for Europe. So we'll have all the regulatory approvals done already.
That's great. So I would pick out a group of physicians, your key opinion leaders, that would be then sort of your clinical trial, field trial people, and then work out some kind of have some interactions with them on Zoom, some meetings where you can decide, here's our initial protocol that we're going with, and then we're going to reconvene at a certain time and discuss our data and then amend our protocol. So sort of adaptive kind of a field test, so that you can then jointly develop what's the best approach to use this thing. And then after that, you could go broader. You can see these guys will become your advocates then.
Yeah, so I think that when we started out, it was the focus was on antigen testing, and there's no issue with antigen testing. People want that; they just want to read it quickly and accurately. So that's how we started out. Right? There's no objections against antigen testing. So we deployed using antigen testing. But I think the challenge is now with antibody testing. That's where the challenge comes in. Suddenly, it's not so easy anymore. to
So and where
do that.
do they stop
Yeah.
it? So like, is it the hospital doesn't want to order the kits, or if machine is already there and it's doing
the machine's already there, the kits are available. It's just a matter of policy if they're going to use it. But there's a big demand for people that they want to test. So we are testing at private clinics, basically. We're doing
I would go with
private events. Yeah,
I'd go with private as far as you can. Get as many private on board. Screw the hospitals for now. If they're just gonna interfere with you, and then you're gonna trigger some media attention, whatever. So I focus on private. Yeah.
So Sasha and Clas, to this point, I think it's a very important point. What would be the best location to kick this off? I mean, I was talking about these 3 physicians, but they are all, of course, in the US. But maybe the place to go is, I don't know, Brazil or something. Yeah, I don't know.
Yes.
And we have connections there too through, the World Health Council, etc. So I mean, but what do you think? Right now the landscape would be most favorable or more favorable in one or the other country?
Yeah, I would, international is always great. If you can get it into the US with a few key people, that's great, because that always carries a lot of weight. So key European, US, and then, Brazil it's a big place. And other countries
with
like
many people vaccinated. With many people vaccinated in Brazil.
Yeah.
Yes. Yeah. So I would just go with, well, first of all, whatever you've got, like who are your best contacts, where your machine's already working, and then figure out, okay, well, I need to add maybe a couple of people in the US for the purposes of, making it more visible.
Yeah.
I think it's another, so like also putting the tests on board that the systems are agreeing with. So you can say, oh, well, you need it for this, right? And then, but it also does these other things. So.
Yeah, exactly. Okay, well, I think we had a very, very constructive, interesting discussion here. So the question always is, what is going to follow up of this? I mean, of course, I'm always, I'm completely behind this. Behind this, I mean, to further explore this possibility in a kind of fast track path, right? But of course, I have to dedicate so much time to so many things right now, so I cannot very deeply involve into this. I can always give input with regard to the immunology, etc., evolution of the virus. But practically speaking, organizing this, I can also contact the physicians. Of course, I can set up a Zoom meeting, for example, with Pierre Corrie, or that I can do, and I would do. But there is many logistics involved. There is regulatory aspects involved. So many other things. So do we have the time, the resources? I think we must. I think if this really works, and scientifically speaking, I think this is really the thing to do. Then I mean, the value that it would contribute is just huge. Just huge. We may not realize it yet because people may still believe, oh, this is not going to happen. But I tell you, I mean, I'm convinced it will. And then we are there not knowing what to do, right?
Well, there's a lot of value in the data that gets generated.
Pardon me?
You need the data.
Yeah, there's a lot of value in the data created because you can use that data to be able to really tell what's going on the ground, right? Particularly when you combine it with metadata of symptoms and vaccination status. That's a very powerful combination.
So how much time would it take to make a kit, for example, that can?
Well, the kits are already out there. I mean, we're testing already. We're deploying and we're scaling, and we're working with test kit vendors to bring more and more tests on board. So Brazil,
does that involve the nucleocapsid? Does that involve the nucleocapsid? Does that already involve the nucleocapsid?
No, I don't think there's test kit, but that's something we need to talk to the test kit vendors about. There's actually a lot of different test kits. Abbott has one. There's ASK.
I know.
There's a lot of them have test kits already. We just need to read them quantitatively and get them into the system.
Yeah.
Yeah. So
Is this something you would do, Clas? Is this something you can
yeah,
start to
I think
explore?
yeah. So because I'm in Norway right now and at the NTNU, the University of Science and Technology. But when I return to Taiwan, we work with the test kit vendors that are there, and it's at least 20 test kit vendors there that are that's their full time institute. Yeah.
Because in terms of resources, I mean, we can. It's also not my core business, but people like Steve Kirsch, etc., could easily put money into this, right? I mean, that should not be an obstacle. On the other hand, I mean, we cannot ask, of course, your company to all of a sudden prioritize as much as I would wish to, on this. But so I don't know really. That are the kind of aspects that I have no idea about.
Yeah. What do you think, Sasa?
Well, I have a bunch of investors that have worked with us for years. Now it's so I also need to understand this is you already have a company. This is inside your existing company, or this is going to be a new venture?
No. All the business side is already structured. Yeah, we have a LLC in the US. We have a Singapore entity. Everything is set up properly. We have 5 investors already from Amazon, Google, and Microsoft. So
I have investors, but they prefer to work with C corps, S corps. LLC is not quite. So, but I have a bunch of investors, the ones that funded us for years, and we have access to others. So if that becomes, necessity of some sort, I can reach out and help you, with introductions.
Yeah, for the U.S. market in particular, we don't really have. I mean, it's the strategy, right? What kind of strategy do we take on? Because in the U.S. you need a lot of you need a good distributor. You need a good.
Daniel left us, but he told me in advance that he would also be interested in helping us with that kind of aspect, right?
Yeah.
So I don't think that should be an obstacle, but it need to be clearly mentioned and clearly stated so that we know what to do. I don't know.
There's a lot of things you can see in the data. For example, high zone tolerance. You can see the amount of spike IG post-vaccination, how it gets reduced over time. Is that as a potential indication of high zone tolerance? You can see efficacy of different vaccines. For example, you see the IgA, which is a harder test case to develop, but immunoglobulin A from mucosal antibodies, very interesting. In natural infection, that develops first, obviously, and then later comes IgM and IgG. Whereas in the vaccination, you have IgM and IgG first, and then later IgA. So there's a lot you can read out of that data.
But this I'm talking about, IgG would just be more than sufficient, right? Just IgG, right? So it could be very simple, just discriminate nucleoprotein or spike alone from spike. Pardon me?
The challenge we faced with IgG is that the various test kits that were developed at different times test differently depending on when the infection happened. So, for example, some of the Abbott test kits they were very early on. They worked great on original Wuhan wild type because that's the time they were developed. And some of the newer test kits are more they give a higher positivity rate for Omicron. So it's even variations. I've found that sometimes they will use 4 or 5 test kits together in one as one panel for an individual to get a full. You can kind of see what they have, what kind of antibody protection they have. It's quite interesting.
Well, that's a very good point that I forgot to say. But of course, those who really still have some natural immunity left will only show that natural immunity, towards, of course, a relevant antigen from the virus they have been recently infected with. So that is very, very clear.
Yeah.
Whereas, of course, the vaccinees, they will develop primarily against the antigen. Of course, they have been primed with, which was in the vaccine. So these are things that indeed need to be taken into account, and it may well be that 2 antigens may need to be tested.
Yeah.
But,
So you really need to get multiple samples per patient. See,
multiple samples, exactly. That's how we dealt with the electrocardiogram issue.
Yeah. Right.
We were reading. We replaced manual reads with automated or highly automated. We still had humans doing quality control, but we took, instead of 3 cardiac beats, we were taking 100,000, and that gave us the precision that we needed, and so. That's how we. So yeah, if you're doing something more automated, you need much more sampling than you were doing before. And I myself don't totally believe in fully automated. You will need some sort of quality control, some statistical quality control process to tell you which results to look at by human.
Right, and we see that too when we develop quite a few tests in Brazil over the past couple of weeks and months, and you can see the human error sneaks in, so you have to be careful about that.
Yes.
Yeah.
Yes, and it's for population. We were doing population studies. It's much more problematic than diagnostic of a single person. But you still need to think about the variability and reproducibility and all those things.
Yeah.
So, and that typically, if you are automating more, you need more samples, and then you need some kind of way of doing the quality control. By human. So.
It's kind of annoying because a lot of times biology doesn't agree with the mathematical models in the lab.
It never does. Biology doesn't work like mathematics. So yeah, that's a lot of engineers come in and like, I can automate this data. I'm like, no, you can't. So
anyway, if you need input with regard to the antigen, I think I can help there as well.
Okay.
Because we would need I don't know whether the antigens used are full antigens, full S protein, for example, or is only part of the protein, etc. All this is important.
So what is your biggest need right now?
Is this a question to me or to Clas?
Clas, as far as his technology, or both of you, if you're like working on this.
I think for Geert, it would be data, right? And my job is to collect the data.
Yeah, well, data, first of all, to see whether this makes sense. Scientifically, for me, it's very, very clear that this makes sense, but we need to see how it pans out in practice, and whether we can really very clearly discriminate between somebody vaccinated who still has some natural immunity left, somebody who hasn't, and then, of course, the unvaccinated who should have full, natural immunity left. So, but the most important thing for me at this point would be: is this feasible? Is it feasible to do so? And does this allow us really to discriminate? Not for the sake of discrimination, but because it may have a huge prognostic value in terms of the feasibility of treating people when they get a disease, right?
Yeah. So feasibility study, get like 3-4 physicians to use it and do your feasibility assessment.
So like I mentioned, we're doing testing in Taiwan, India, and Brazil right now. Those are main focus areas. So I think an ask would be suggestions on the strategy for Europe in particular. Geert, how to deploy in Europe. We have mass testing sites in Germany right now.
Yeah, it's a shame that Christoph couldn't join. He must have forgotten about this, or maybe because he's from Germany and he's very, very active, has a huge network. So he would be really the guy to ask, right?
And in the US, so how do we deploy this in the US? We need to find the right talent in the US and scale.
Well, I know there's people in the US, and with regard to the logistics or some practical thing, I think Daniel could help a lot. He's very engaged and motivated to do so. So yeah, I mean,
Yeah,
please.
there are also some groups on Telegram or Zoom. Also, they meet every week. There's Medical Doctors for COVID Ethics group. I can introduce you, Stephen Frost. He's in the UK, I think, and they typically have 100 people on Zoom every week, and a lot of them are physicians. Not all of them are physicians, but a lot of them are, and a lot of them are. Well, also in the US, we have naturopath practices as well. So, and those are independent, and they will, they're looking to treat vaccine injured, and they're looking for tools. So you could arrange a meeting, a presentation to the group and say, here's what I'm working on, and I'm looking to sign up some people into my feasibility, exercise, field test, and, work out some kind of relationship with them. But yeah, that would be a way to just go and reach out those people who are independent and they're looking for these things right now.
Yeah, it's good advice. Thank you.
Okay.
We started the meeting by talking about the grand state of the universe, kind of. Geert, are you familiar with Matthias Desmet? He probably went to the same university. Does he live down the street from you?
Pardon me? Yeah.
Does he live down the street from you? Pretty much.
Well, yeah. Well, Belgium is a small country, right? Yeah. I mean, we see each other on a quite regular basis. He's of course also extremely busy, and
mass formation.
Mass formation. Yeah. Well, I mean, the thing that we share is that we both of us think at the population level. I from a viewpoint of the evolutionary dynamics of the virus in a population, and he, of course, of the psychological, the psychological impact of this on populations, right? So yeah, it's interesting. But he has a very analytical mind, and he can just express this in I think a very, very, very special and speaking way, so that people feel all this anger and this stress and these weird things, but he can phrase this in words that people really say, oh yeah, that's exactly what I meant. That is what I feel. That is he does this very well. Yeah.
Great communicator. Are you familiar, Sasha, with?
Yeah,
I'm a big fan. Please say hi.
Yeah. I will.
Awesome.
So, Clas, I mean, my proposal would be to have regular follow-ups, maybe shorter meetings. I mean, this was first meeting, a lot of things to say, to explain, but maybe we can have some follow-up meetings, shorter, to the point, just discussing some practical things. We should involve, I think, Christopher Plot. It's not good to have meetings with 20 people either, but I think 4 or 5, maximum 6 people would be good. Sasha, are you interested also to further support this in one way or the other, or?
Well, if I can,
because it's.
Yes, I mean, I'm happy to help, make introductions or provide some advice. Yeah, feel free to include me if it's, if you want to have a smaller group or whatever. But like, when you think that you need my advice, feel free to involve me, and I can introduce you to these groups. And maybe you can go talk to physicians all over the world through these channels, and see if you can get some people on board. So that was always my approach. Try to sell it to a couple of people first, and see how they use it. But it was very nice meeting you, and thank you for inviting me. And you have my email. Feel free to.
We're supposed to have a talk about the children. Is that I think we didn't really get to that focus. Maybe we should schedule another one, particularly to discuss immunology in the children.
Let's do that maybe another time, because this is pretty complex. It really comes down to the fact that if children are having this highly antigen-specific vaccinal antibodies, whether they acquire them actively because they get vaccinated themselves, or they get them passively from the mother through the maternal antibodies. It's a pretty complex phenomenon, but in fact, these specific antibodies at the early stage of their life is going to, in fact, outcompete, because they have higher affinity for the virus. They are going to outcompete innate antibodies. So they will prevent innate antibodies from binding to the virus. And that binding of the virus, of live virus, to the innate antibodies is critical for the newborn to make that switch from passive immunity, where it is protected by the mother, in the womb, and later on by maternal antibodies, to activating its own immune system. And what that immune system needs to be able to do, like all immune systems, is to discriminate foreign from self. And this, phenomenon, the binding of the innate antibodies to glycosylated components, be it viruses, be it even some bacteria, is critical to educate the immune system to do this. And there are even its own proteins that are produced more and more, of course, as the baby is growing, will be by its body, by its metabolism on purpose be glycosylated, so that they can enter into this process and they will educate the innate immune system on how to induce peripheral tolerance towards its own proteins. And that is thanks to the glycosylation, right? So it's a very complex thing, but one thing is for sure, that you completely disturb this, by vaccinating them in those early days. And that is, by the way, the reason why we are not using inactivated vaccines to immunize children. We are using live attenuated vaccines like rotavirus, like measles, mumps, rubella, varicella. All this stuff is live attenuated, right? So it's very, very important. People know this, and nevertheless, they say all of a sudden, oh, you know what, coronavirus, we are going to do this differently. We are going to use a non-replicating virus. That is what it comes down to, right? So it's very, very complex, but it will be dramatic. Listen, I'm always saying we can stop the mass vaccination tomorrow. It's not going to stop the evolutionary dynamics of the virus. We have pushed it way too far. The only thing we can still do, is to try whatever we can to prevent our children from getting vaccinated, because that is going to be a catastrophe. It's going to be a catastrophe as well in terms of immune pathology, as these children are not being protected against, many other glycosylated viruses, like the flu, like RSV, like even measles, mumps, rubella, if they have not been vaccinated before. But if you first COVID vaccinate them, you can no longer give those vaccines, right? So it's, and not only that, also a number of other glycosylated pathogens. Think about streptococcus, staphylococcus, meningococcal, for example, Haemophilus, etc. There's all glycosylated pathogens that they will not have been educated to recognize, right? So and I've studied this very much in detail, because frankly speaking, in all modesty, there is very, very few people who understand this, very few, right? Yeah. And it's frustrating, because I need to communicate this to the parents, but even experts, even doctors, even pediatricians don't really understand what is happening. But you can imagine, Clas, that it is a very specific thing for a child to make that transition from being completely passively protected by the mother, maternal antibodies or through, antibodies that go through the placenta, etc., to all of a sudden, it's just, on its own, to kick off its own immune system and to teach that immune system to discriminate foreign from self. That is a very important transition that it needs to go through. And we are just throwing a bombshell on that process, right? It just, it's so energetic.
I gotta go, but please keep me, posted and reach out if you need some help. And I will. We will stay in touch.
Okay. Well, both of you, thanks for your time, and Clas. We will follow up on this. Please, keep going, and let's do this. Let's try at least. Let's try. Okay.
Thank you very much.
Thank you. Take care.
See you soon.
Bye bye.
Thank you.
Thank you.
Bye.
Yeah, bye.